Hi, everyone. I'm Akanksha. I'm a postdoc at the Institute for Bioscience and Biotechnological Research at the University of Maryland in collaboration with the National Institute of Technology, Standards and Technology here in the United States. And I would like to start by thanking the organizers for giving me this opportunity to share my research with you and everyone from the scientific community joining from across the globe. Today, I'm very excited to talk to you about the computational arm of my research, which is helping use molecular dynamic simulations or computational approaches in general to accelerate rational drug development for nucleic acid therapeutics. And for this case, we will use antisense oligonucleotides as an example. And in particular, I'll be using the phosphorothioate modification as an example. And I'll hope to convince you that these simulations can be applied more generally and broadly to this new modality of therapeutics. And like I said, this is just the computational arm of my work. I also have an extensive experimental arm on the same work. If you want to know more about that, my contact details are on the left of my slide with my LinkedIn and email address, so feel free to reach out. And with that, let's start. To orient us all, if I can, to orient us all, I'd just like to briefly tell you about RNA therapeutics in general. They are classified into two broad subcategories: the short oligonucleotide therapeutics, which comprises aptamers, siRNAs, and ASOs, as in contrast to the longer category, which is mRNA. Today, I'll be talking about ASOs, as I said, but because all aptamers, siRNAs, and ASOs fall within the general size regime of 15 to 50 nucleotides, everything that I talk about today is applicable to all these three classes, but not to mRNAs. And we all know RNA by itself is a poor drug modality. So to make RNA into a drug modality, we have to extensively modify it on the phosphate backbone, the sugars, and the bases. Here, I'm showing you all the modifications that are present in the currently approved therapeutics. And all of the chemists in the audience know that there is a wide breadth of other novel chemical modifications that we can apply to these therapeutics, and they may actually have much better therapeutic properties. But because this chemical space is so vast, exploring it systematically has practical limitations. It has monetary limitations, it has experimental time, and labor limitations. So our approach to solving this challenge of exploring the chemical space for oligonucleotide therapeutics is computational. In particular, I'm going to be using a computational technique called molecular dynamics. And what molecular dynamics basically does is it simulates the dynamic behavior of biomolecules in solution. And why do I say dynamic? We are all aware that biomolecules are not static, they're dynamic, and their biophysical and biochemical properties, and by extension, their therapeutic properties, are dependent on this dynamic behavior. So in order to be able to capture experimental properties, we do need to simulate the dynamics of these molecules. So what MD does, in essence, is create a movie of your molecule, and my movie is not playing, which is okay. So here, what I have is a movie of an RNA, a hairpin loop RNA, and in gray, you're seeing some magnesiums. You can see some snapshots here in my movie, these ions, and as a result, the RNA moves a lot on a biologically relevant time scale. So what MD basically does, it starts with a starting 3D structure, which can be experimental, determined by X-ray or cryo-EM, that you can download from the PDB. Or if you don't have a structure, you can generate a model using, for example, AlphaFold. And this, you can define the structure and energy of this starting structure. What you can then do is model each of the bonds as a force, because that's essentially, when simplified, that's what a bond is. It's a force between two atoms. You can compute the force, apply Newton's equations of motion, move each atom based on these forces ahead in time, and then you generate a new configuration, a new position of all these atoms in space, and you iteratively keep doing this on a biologically relevant timescale to generate this movie of your molecule. What you can then do is analyze all of these snapshots and look at different structural properties and try to correlate them to something that has been experimentally measured. So in order to do molecular dynamics simulations, an important component is to compute the forces. How do we do that? Is we essentially have to completely describe every atom in your simulation. So every atom in your nucleic acid needs to be described. You need to define properties like charges, their van der Waals radii. Then you also need to define all your force constant between different bonds, different dihedrals. So as you can imagine, this is a lot of parameters that you need to define. While these parameter sets are very easily available for native nucleic acid residues, ACGT, they are not so readily available for a modified nucleic acid. And that's why molecular dynamic simulation hasn't been so widely applied in the field of oligonucleotide therapeutics. A recent advance by our team and that I'm highlighting over here, which comprises both academia, government, and industry, we all joined forces and came up with this program called modXNA, mod standing for modified, which generates over 100,000 chemically modified nucleotides in an easy-to-implement manner and is also extremely high throughput. That means you can do simulations of oligonucleotide therapeutics with any kind of modification that you can think of on scale. And you may ask, hey, how does modXNA does generate these very chemically diverse libraries? It's because it's very modular in nature. It combines a phosphate backbone modification with a sugar modification and a base modification. So you can choose any combination available in the catalog to generate any modification that you can dream of. And then you can use that to run your MD simulations. To learn more about the original modXNA paper, look at this 2024 paper. And if you want to learn how to use modXNA, we recently published a tutorial in this 2025 paper. And more importantly, the catalog of these modifications is constantly expanded. If you have any requests, feel free to send it to us, and we can try to generate force fields for you. So what our hope is using modXNA, this very powerful program, is this. We hope that we can take a bunch of novel modifications. They can be on the order of hundreds, 500s, whatever you wish. You can generate parameters for these, the force constants that I showed you in my previous slides, run MD, and using that, you can analyze the structural properties and look for properties that are therapeutically advantageous. That gives you some hits that are optimized by MD. And then it's experimentally much easier and feasible to take a small number of hits and test them experimentally for better therapeutic properties, in contrast to scanning this wide chemical landscape. In order to accomplish this dream, we first have to establish that parameters generated by modXNA and used by molecular dynamics simulations actually reproduce something that has been known experimentally for these modifications. What that essentially means is that I need a test case and I need to benchmark this pipeline, which is what the rest of my talk is going to focus on. And I'm going to use phosphorothioates as a test system for these simulations. Phosphorothioates, as we are all aware, are a very common modification in the field because they provide nuclease stability, which is an important property, and they also enhance cellular uptake of these negatively charged RNA molecules by the cell. In addition to these favorable properties, phosphorothioates also generate a challenge, and that is a challenge of stereochemistry. When you have a phosphate nucleotide like that shown here, both of the atoms on the phosphorus are oxygen, so this is an achiral atom. When you change one of them to sulfur to make it a phosphorothioate, you have two possible configurations of the sulfur. In one, the sulfur faces the major groove, that's called the all-P configuration, and the other configuration where the sulfur faces the minor groove, which is called the SP configuration. A number of studies have now shown that the orientation of the sulfur affects all properties of the therapeutic, such as efficacy, safety, durability of response, telling us that this is an important property to measure, understand, and control in these therapeutics. So I will do just that. I will use test system, which is Tegsedi, an antisense oligonucleotide therapeutic that is entirely modified with phosphorothioates on the backbone, like that shown here. And we will duplex it with its native mRNA counterpart that would be encountered inside the cell. This is the whole schematic of the hybrid duplex we will be looking at. I would like to point out here that the question we are asking MD to solve is very difficult. We are just changing the orientation of one little atom on the backbone and asking MD to reproduce everything we know experimentally about R and S diastereomers for this complex modification. So if MD can pass this test, I have some confidence to say that it will be able to do well for novel modifications. Before we move further, I would like to highlight that this hybrid duplex that we are using contains a DNA backbone in the middle, shown in pink here, hybridized to an RNA backbone here. And before I move to the results of these simulations and tell you how amazingly MD simulations do, I'll switch gears to talk a little bit about the geometry of DNA and RNA helices, and in particular, helices that are formed by combining a DNA strand and an RNA strand, because that's exactly what we are encountering here. So as all of us are aware, both DNA and RNA form double helices. When DNA combines with DNA, it forms a B-form helix, which looks something like this. When you look down the helical axis, you see that there is no cavity in the B-form DNA helix. It's very nicely stacked and fully packed. When you look at an A-form helix, which is formed when two RNA strands come together, and you look down the helical axis, you see this nice little cavity in the middle of the aRNA helix. So these geometries are very classically defined for bDNA and aRNA. And when you start to look at the conformations or geometries of individual nucleotides, particularly the sugars, you see that in DNA, the C2' atom here faces the same, is on the same phase as the nucleobase. We call this conformation C2' endo or south. This is very typical of B-form geometry, whereas A-form geometry has a different sugar conformation where the 3' atom is facing the same side as the nucleobase. We call this C3' endo or north pucker. It will be important for you to remember what C3' endo refers to, because it will keep coming up later in the slides. So now you can ask, hey, if I combine a DNA strand and an RNA strand, what does it look like? Does it look like an A form or does it look like a B form? Well, in most cases, a DNA-RNA hybrid looks more A form-like. That means the DNA adopts more A form-like geometries in a DNA-RNA hybrid. So before I move on to doing my simulations, I need to first ensure that I'm choosing parameter sets that allow the DNA in the Tegsedi ASO to sample RNA or A-like conformations, because experimentally we do know that is true. So to do that, I went ahead and tested some force field parameters that are commonly used in the field for DNA. I'm showing you 3 here. And then I am looking at the movies of my molecule and counting the number of frames where the DNA adopts an RNA-like north pucker conformation, shown over here. And that's what we are seeing here for the different base pairs in my little toy system for these 3 parameter sets. As we can clearly see, only OL24, which is depicted in red here, allows your DNA to sample a north pucker or an RNA-like conformation between 20 to 60% abundance. The blue and the black do not do so well. So we went ahead by using OL24 for all our simulations of Tegsedi mRNA duplexes that I will show in the next slide. The next question I'm going to ask is whether MD is able to reproduce experimentally observed thermodynamic properties of these oligonucleotides. It has been shown, like I'm showing here one example from the literature, that if you modify your DNA with a fully RP backbone, it shows a higher stability or TM when it hybridizes with its RNA counterpart as compared to an SP. This is a well-known fact in the literature since the 80s. So the first question I asked, hey, if I do my MD simulations with my RP modified Tegsedi and my SP modified Tegsedi hybridizing to the same mRNA strand, am I able to reproduce these thermodynamic differences? And voila, I am. Here I am showing you the hybridization enthalpies of an RP duplex and an SP duplex. We see that the RP-PS ASO binds its target mRNA with about 5 kcal per mole higher thermodynamic stability as compared to the SP, giving us the first evidence that modXNA and MD simulations are able to generate parameters and trajectories that are able to recapitulate in a quantitative fashion properties that have been known experimentally for years now. Now that I have answered or benchmarked the system a little bit, we can have fun. We can start asking questions like these. Why is the RP duplex more thermodynamically stable than the SP duplex? And here I would like to point out that to the best of my knowledge, this question hasn't been answered in the literature before. To do this, I took a structural and dynamic approach. That means I went ahead and dived deep in the movies of my molecules and I started analyzing their structural properties. Here I'm showing you one example. So the movement of nucleic acid base pairs can be described in terms of certain parameters. You can think of them roughly as some degrees of freedom. I'm showing you a set of degrees of freedom here that we call the base pair step parameters, which is basically defining the movement of every base pair step. So every 2 base pair steps, how are they moving with respect to each other in each of these translational and rotational directions. So when I look at how these parameter values vary for RP and SP in that entire movie of these molecules, I'll take one example here. If I start to look at this twist value, which describes how much of a base pair twist as compared to the other in the helix, if I plot the distribution of all the twist values that the SP and the RP duplex sample, we see that these distributions look very different. If you look at the RP, it's a very tight distribution. What that essentially means is that the RP duplex isn't moving much. It's a nice little rock, and it stays like that during the simulation. SP, on the other hand, makes a lot of excursions and shows a greater variation of twist values. It's doing a lot of things in the simulation. You can look at one other example that I used here. It was tilt, same distribution. RP is a rock, SP moves a lot. What this helps me conclude that, and also in addition, if you go look at what these structures look like in these variations and this rock, in the RP rock, I find that the RP duplex just looks like a stable A-form helix and it does not do much. All these excursions that the SP is making, they are towards the B-form helix, telling me that the SP has more propensity to form the B-form helix as compared to the RP. And this will become important later. In addition, we know by these box plots, I was able to conclude that SP is more dynamic as compared to RP. I can do some fancy mathematical manipulations with these data and try to define the rigidity of each of these base pairs. So which is basically quantifying this variation that I'm showing you in the parameters here. And I can do it for all of these 6 parameters for each of the base pairs in my helix to further quantify these rigidities that I'm intuitively seeing from these plots here, which is exactly what I did. I calculated the stiffness constant for all these parameters or degrees of freedom using this mathematical equation shown here. I can go into the detail during the discussion. In the interest of time, I'll just tell you that you can calculate the stiffness constant for each of the base pair steps. And when I go ahead and do that and plot it, so here, these are the 6 degrees of freedom, the 6 plots. On the x-axis, I am plotting the stiffness constant of the SP duplex. On the y-axis, I'm plotting the stiffness constant of the RP duplex. The dotted line is the y equals x line. And everything which is above the y equals x line tells me that the RP duplex is more stiff as compared to the SP duplex. And we see a majority of the base pairs in all of these degrees of freedom, the RP is more stiff than the SP. Previous studies have linked this stiffness of these modifications to their stability. This paper in 2009 very nicely showed that if you look at the stiffness, which is on the y-axis, and the melting temperature, which is stability on the x-axis, they very nicely correlate. suggesting that things that are stiffer are also more thermodynamically stable. If I combine this fact with my finding that RP is more stable, that RP is more rigid as compared to SP, I can conclude that the higher rigidity of the RP duplex potentially provides a dynamic explanation for its higher stability. And again, to the best of my knowledge, this is for the first time we have understood why RP duplexes are more stable as compared to SP duplexes. And that brings me to the conclusion of my nucleic acid story. I know a lot of you have been holding your breath for the enzyme, which is the other actor in the story, RNase H. So let's do simulations of these molecules with RNase H. That's exactly what I did next. Here, I was lucky that there is a structure of human RNase H in complex with a DNA-RNA hybrid that I'm showing you here. A first glance at these structures helps you recognize that this helix isn't fully A form or B form. It's something in the middle. If you look at the cavity, it is not as wide as what an A form would give you, and it's not as narrow as what a B form would give you. You can further quantify the A or B formness of these helices using the same sugar pucker that I showed you. Here, I'm plotting the sugar pucker angles of the RNA residues, and we see that they are a nice little cluster, which is very A form-like. The DNA, on the other hand, is very B form-like. So in this complex structure with RNase H, the DNA is actually not becoming A form-like, which is what we were seeing was happening in the absence of RNase H. So this gives us some hints that the RNase H is imposing a B form-like conformation on the DNA. If you remember from my rigidity slide, we saw that SP has a higher propensity to go to the B form. It shows excursions to the B form, whereas the RP does not. And if RNase H likes to keep the DNA in the B form, when it encounters an SP, it will not have to waste any energy to make it B form because the SP itself wants to go to B form. So all the energy that the RNase H is putting into the system is going to go towards favorable binding energy. And that brings me to my prediction. I predict that because SP has a higher preference for B form DNA, it would show a higher binding affinity to RNase H. So let's test that. To do that, we have to first do the simulations of these molecules. And again, my movie does not play, for which I apologize. This is a movie of a DNA-RNA hybrid, the same structure that I showed you in the previous slide in complex with RNase H. And what essentially happens in this simulation is that the two parts of the molecule of the hybrid, the part that's contacted by the RNase H, and the parts that free moved almost independently. The part that's contacted by the RNase H does not move a lot. The other parts do. You can take this entire trajectory and do some post-processing to extract the binding affinity of the RNase H to this hybrid duplex, which is what I did. But, oh, it actually plays. So yeah, here you can see independent movement between this part and this part, which doesn't move a lot. So next, I did the post-processing of this entire trajectory to characterize the binding affinities. And before showing you my results, I would like to point to this paper where they did experimental measurements of the cleavage activities of human RNase H, the exact same that we are using it in our simulations over here. And they found that all SP backbone, where all the linkages are SP, is a better target for RNase H as compared to all RP. Here you can see that in the cleavage assay, all RP, there is a lot of all RP product left at the end as compared to all SP. So we do know that SP gets cleaved better by RNase H. If we make a simple assumption that cleavage, that binding affinity is a proxy for cleavage, which is not the best assumption to make, but in these cases, this is the data I had available, so I had to compare to that. So what I did next was compared binding affinities obtained by MD to these cleavage efficiencies. What I essentially did was generated two duplexes, an RP duplex and an SP duplex, and I combined it with RNase H and calculated this binding affinity that I'm showing you here. And as predicted, also as observed experimentally, we do see that SP has a more favorable binding by about 20 kcal per mole to RNase H as compared to the RP counterpart, suggesting that, showing us some evidence that the RNase, because SP has a better affinity, has a better ability to form the B-form conformation, it binds tighter to RNase H. And this is another example where modXNA and MD are able to reproduce experimentally known parameters in a quantitative fashion. That brings me to the conclusions and future directions for this project. I hope I've shown you that SP and RP phosphorothioates, I apologize for the spelling error here that missed me, they exhibit very distinct structural and thermodynamic properties, and more importantly, MD can recapitulate these. And because now I've shown you a test case and a very tough test case where I'm just changing the orientation of an atom, and I've shown you that MD can successfully provide very relevant therapeutic answers, I hope this pipeline that I showed you earlier in my slides, where you can use modXNA and MD to screen a bunch of novel modifications to find optimized hits, which can be further experimentally validated to accelerate rational drug development in the field of oligonucleotide therapeutics. And with that, I would also like to acknowledge my mentors, Christina and Rob, who have been instrumental in the overall progress of this project. Christina specifically for her immense guidance on molecular dynamic simulations. I would like to thank the entire modXNA team for developing this wonderful program that can do all of these very complex things in a very easy manner. All my collaborators for the wider project on ASOs, other postdoctoral associates, my friends at IBBR, graduate students that I've had the privilege of working with, the administrative and ID staff at IBBR, the FDA for funding, and all of you for listening in. With that, I will take any questions here, and if we don't get to your question, please feel free to email me. Over here, this is my LinkedIn. I'm currently looking to transition in industry. So if you would like to network, also please feel free to reach out. Also, if you want to learn more about my experimental work, feel free to reach out to me. Thank you.
Thanks a lot for this excellent presentation. I really enjoyed it. We have time for a very few short questions since we have a very busy schedule today. So if you have a question, then please use the Q&A function. I think so. My first question would be: so how to use this for preclinical researchers that actually want to develop a compound to screen in their models of interest. I think many of us have like a disease of interest. They have like a target of interest, and they already developed a few compounds. But how can you use this technique to develop a better molecule to be tested in your preclinical and eventually clinical models?
Great question. So just to make sure I understand the question correctly, when you say you have a molecule, that means you have a sequence that is...
A sequence, correct.
Yeah, that has been optimized. So now the challenge is what modifications you should put on it to make it a good therapeutic target. How you can use that is exactly where modXNA would come in. You can start with modifications that are in currently approved therapeutics that I showed in my slide. And you can put them in any position that you want. You can try any combinations you want, and you can simulate them using the techniques that I showed you, and look which one of them give you better binding affinity, give you more rigidity, like the ones that I showed you. Or you can go ahead and, if you want to be creative, try some new modifications. You can use modXNA to generate its parameters. For example, you want to try a locked nucleic acid, a PMO, a PNA. modXNA, it's once you install it, it's just one command line statement that you have to write, and it will generate everything you need to simulate that. What you have to then go ahead and do is take your 3D structure file, let's say a PDB file, and you have to change whatever nucleotide you want to make into a modified nucleotide by the modified nucleotide that you just generated, and you have to submit it to an MD simulation. It's as simple as that. So like generating modified nucleotide will take you 30 seconds, which is extremely fast. And you can also make automated pipelines, which is something I have done, that will just make a bunch of modified nucleotides for you by the click of a button. And then you can also automate the molecular dynamics simulation pipelines itself. So all of this can be done in a parallel fashion. The only limiting factor is how much computational power you have. If you have computational resources, you can simulate many things at the same time.
And then the follow-up question, of course, is, have you already tried this in the lab?
So that is the next step of the project. The first was to benchmark the system, which is exactly what we were able to accomplish here using phosphorothioates. Yes, so the dream is to next go to novel modifications, simulate them, find some hits, and then experimentally validate them. For that, obviously, we need a lot of collaborators. So you or anyone else in the audience who would be interested in this, we'd be very happy to collaborate on all the experimental arms of this project, which are extremely essential.
Well, I think there are probably many people that want to make use of this molecular dynamics strategy. Yeah, good. Thanks a lot for your presentation. I think
Thank you.
since we have three, we need to move on to the next speaker. So I think give the word to Erin.
Great. So our next speaker is Arnau Dominguez.
Okay, let me see if I can share the screen. Oh, one second. Okay. Can you see the screen? Yes, right?
Yeah, perfect. Yes, we can see the screen.
Okay. Perfect. So
Correct.
hello, everyone. My name is Arnau Dominguez. I'm a fifth-year PhD student from the Institute for Advanced Chemistry of Catalonia. And today I would like to tell you about the project that I presented at the last OTS, where we try to understand the role of C2' fluorine substitutions in parallel triple helix structures. So first of all, I would like to start giving a brief background about triplexes and how we can use them for therapeutic applications. So triplexes are formed in polypurine-rich sequences by the interaction of three strands, two of them that bind through Watson-Crick hydrogen bonds, and a third one that receives the name of triplex-forming oligonucleotide that interacts with the major group of this double helix, forming Fuchstin or reverse Fuchstin hydrogen bonds. Depending on how this TFO is accommodated, triplexes are classified into two main categories: parallel triplexes, which in general form isomorphic triads, and antiparallel triplexes, which form non-isomorphic triads. As you can also see, for the formation of one of the triads of parallel triplexes is needed the protonation of the amino N3 of cytosine, so these structures are going to be more stable under mild acidic conditions. So the next question here is how we can use triplex formation for therapeutic purposes. So the traditional approach that has been used during the past years consists of designing these TFOs to bind to a promoter region of a specific gene, thereby blocking polymerase binding and inducing the inhibition of gene expression at the transcriptional level. So by contrast to other therapeutic strategies, here we are directly targeting the genomic double-strand DNA, which can be useful for targeting highly expressed genes, for instance. Triplex formation can also be used for targeted mutagenesis to induce DNA damage and also for epigenetic editing. In this case, what we do, we couple a mutagenic moiety to this TFO, and when the TFO binds to its triplex target site, it can induce a double-strand break that can later be repaired, for instance, for homologous recombination. In addition, we can also couple these TFOs to one of the two Watson-Crick sequences, generating these hairpin or clamp oligonucleotides that can also target these polypurine-polypyrimidine sequences through strand displacement. And they can also be used to block gene expression at the transcriptional level. So despite all of these applications, triplexes face several challenges which have hindered their full implementation in real applications, and can be summarized in the following points, which are that triplex formation is always limited to these polypyrimidine sequences, so we cannot target any specific gene. Also, the DNA accessibility within chromatin that can difficult sometimes the binding of the TFO with its target. Also, the pH sensitivity of parallel triplexes, which, as I mentioned at the introduction part, they are more stable under mild acidic conditions. And lastly, the inherent low stability of triplexes under physiological conditions due to the close proximity of the negatively charged phosphate backbones. So to address all of these challenges, here in this work, we selected this intramolecular parallel triplex model containing two hexaethylene glycol linkers, and we incorporated two different types of C2' fluorine modifications, which are FANA and fluororibose, not only to stabilize the parallel triplex under physiological pH conditions, but also to study which are the molecular interactions that can stabilize this motif and could be used in the future to design other chemical modifications that could further stabilize parallel triplexes. So why we selected these C2' fluorine substitutions? As probably most of you know, because they are commercially available, they offer this nuclease resistance that is that important for therapeutic applications. They have also been shown to induce a thermal stabilization in duplexes and other non-canonical structures. And most importantly, they can also be used in fluorine-19 NMR spectroscopy, as we will see later. So what we did here, we fully modified both the polypyrimidine strands of this intramolecular parallel triplex model. First of all, we modified the strand involved in the formation of the duplex, which is the one inside the red box. And later, we also modified the other polypyrimidine strand involved in the formation of the triplex, which is the one inside the green box. First of all, what we did, we assessed the formation of the expected parallel triplexes using different biophysical techniques. We performed CD spectroscopy and also proton 1D NMR experiments. Most of the chemically modified oligonucleotides displayed the typical CD profile of parallel triplexes, which is characterized by this maximum between 260 to 280 and another minimum at 210 nanometers. So they also displayed these immunoproton signals characteristic of protonated cytosines, that most of them disappear at 25 degrees, in consistency with the lower stability of the Hookstein sequence. After that, what we did, we assessed the thermal stabilization of each one of the modification patterns using UV melting experiments and also melting experiments using proton 1D spectroscopy. What we observed here was that the FANA substitutions were always stabilizing the parallel triplex at pH 7 when they were both located at the Hookstein and Watson Crick strands. By contrast, the fluororibose substitution was only stabilizing the parallel triplex at pH 7 when it was located in the Watson Crick strand. So here what we have is an opposite stabilization trend of these two C2' fluorine substitutions when they are located in the strand directly involved in the formation of the triplex. So another interesting thing that we can do with these fluorinated oligonucleotides is that we can track both the unfolding of the duplex and the triplex using fluorine-19 spectroscopy. What we do here, when we incorporate these C2' fluorine modifications in the Hookstein strand, we can track the unfolding of the triplex. And by contrast, when we modify them in the Watson Crick sequence, we can follow the unfolding of the duplex. This technique could have really interesting applications in the future for the, for instance, for designing fluorinated TFO proofs to study triplex formation in cell. Also, they could be used to investigate for high affinity triplex binding proteins and also to study cellular processes where triplex are involved. Finally, to study the molecular interactions that could explain these opposite stabilization trends, we performed these 2D noise experiments that I won't go into much detail here, But with that, we are able to obtain these solution structures that we can later use to explain the stabilization trends that we previously observed. So here we have the solution structures that we obtain for the parallel triplexes that have been modified in their Hooks-Dean sequences by both FANA and fluororibo. And the double helix that they form are in both cases a mixture between an A and B type. But if we take a closer look, we can observe that the FANA substitution is pretty well accommodated within the parallel triplex and can form this hydrogen bonding network that stabilizes the structure. By contrast, when we compare it with the fluororibo substitution, we can observe that the polarized H2 proton and the fluorine atoms are in the opposite orientation and can only form few stabilizing interactions. By contrast, when we incorporate these C2' fluorine substitutions in the Watson-Crick sequence, which is this one here that forms the duplex, we don't observe any specific interactions with the Hooks-in strand that could explain the stabilization interactions that the stabilization trends that we observe. And by contrast, what we believe that is happening is that the thermal stabilization that these both the FAN and fluororibo are imparting to this to the double helix is pre-organizing the duplex and is facilitating the accommodation of the third Hooks-in strand, thereby stabilizing the parallel triplex. In addition, also the inductive effects of these fluorine atoms could be polarizing the triads and stabilizing them. So to conclude, what we have observed in this work is that indeed C2' fluorine substitutions can stabilize parallel triplexes and could be used in the future to design fluorinated TFOs for all the therapeutic applications that I mentioned at the introduction part. Also, when these C2' fluorine substitutions are incorporated in the Watson-Crick sequence, they are always stabilizing the parallel triplex at pH 7, and by contrast, only FANA is well accommodated within the Hooks-in strand. Lastly, the fluorine-19 NMR spectroscopy technique that we have shown can be used to follow the unfolding of the fluorinated sequence and could be used in the future to study triplex formation in vitro and potentially in cell. So that was all. I would like to thank all the people that made this work possible. Of course, all of you for your kind attention, and I would be happy to take any questions.
Thanks, Arno, for this presentation. I think when I think about these kind of modifications, I always think about Masad. So I see you also collaborate
Yes, we are
with him,
collaborating.
because I think he's always very keen on promoting these kinds of substitutions. And actually, that
Exactly.
also brings me to my question. So, what about other kind of modifications? So, of course, you have now, I think, fully focused on these.
Yeah, the good thing with these C2' fluorine substitutions is that we can use the fluorine-19 NMR spectroscopy. So it's a good way to study all of these triplex formation in cell on using all of these techniques. But we have also worked with other like 2' O-methyl and all of kinds of medications. So yeah.
And so, are you also considering to test any other single molecule techniques? I think you have this optical tweezers strategy. So, are these things that you are considering in your research?
Yeah, at the moment we are just focused on NMR and other biophysical techniques, and we haven't thought about that. But yeah, for sure, would be another technique to use to further characterize this. Yes, for sure.
And also, I think the same question as the previous talk. So, of course, I think this is a super important research, but this is really at the beginning. So, how to translate this into a more like a preclinical setting where you really use your gene of interest or a sequence of interest. So how big is this step and how to translate this?
Well, yeah, the biggest step I would say is the sequence limitation that, as I mentioned, we are always limited to these polypyrimidine sequences. So it would be also the next step would be to find a good target that is able to form these triplex structures. And right now we are not working on that, but there are like other collaborators that are trying to like improve these designs and trying to target all of these sequences that you just mentioned. Yes.
Cool. Well, thanks a lot. I think I give the word back to Erin.
Great. Okay, now we have our last speaker. Keep in mind, we'll stay a few minutes after the hour in case there are questions. And so thank you for your patience in advance for that. Our next speaker is Ophélie Vacca. So you can go ahead.
I'm going to share my screen. Okay, can you see it?
Yep, that's great.
Yeah, it's okay. Good. So hi, everyone. First of all, I would like to thank the OTS for giving me the opportunity to present part of my work today. So my name is Ophélie Vacca. I'm a research scientist working in Aurélie Goyenval's lab at University of Versailles in France. I'm focusing on developing innovative RNA and gene therapy approaches, including exon-skipping strategies, gene therapy vectors, and central nervous system delivery methods to improve dystrophin restoration in Duchenne muscular dystrophy. So today I'm going to present you my results on the neonatal delivery of exon skipping. Sorry, I couldn't see all the screen. Okay, good. First, a brief introduction about dystrophy, Duchenne muscular dystrophy, which is an X-linked recessive genetic disorder affecting about 1 in 5,000 male births with a life expectancy of 30 years. DMD is caused by mutation in the DMD gene, and depending on the mutation position, as you can see here, one or several dystrophin can be affected, causing a progressive muscle degeneration, and for 50% of patients, brain comorbidities as intellectual disability, autism, attention deficits, anxiety, and obsessive compulsive disorder, indicating a pressing need to address central nervous system pathologies and therapeutic approaches. In the brain, you have 3 different isoforms expressed. You have the DP427, the DP140, and the DP71. And also in the gene, you have a hotspot between exon 45 and exon 55, leading to the loss of 2 isoforms, the DP427 and the DP140. And to study that, we have a murine model, which is the MDX52 mouse model. So our general objectives is to assess the impact of postnatal restoration of dystrophin expression as on neurobehavioral features in dystrophic mouse model using exon-skipping technologies. We would like to explore how effective are genetic therapies in the brain, and we also would like to know what deficits can be rescued postnatally. So the MDX52 mouse displayed autistic type disorders as enhanced anxiety responses, fear response, learning and memory disorders. The MDX52 mouse has a deletion in exon 52, leading to a disruption of the reading frame between exon 51 and exon 53. And the translation stopped prematurely, and we obtained a truncated and non-functional protein. So our strategy is to use the ASO in order to skip exon 51 to restore the reading frame between exon 50 and exon 53. and to obtain an internally deleted but functional dystrophin. So we already published some results with ASO exon 51 strategy in adults. And we, in this study, we injected 8 weeks old mice, and we obtained partial DP427 restoration between 3 to 10% in the cortex, hippocampus, and cerebellum. And this partial restoration improves anxiety in MDX52 mice, in the light-dark choice test, and also in the elevated plus maze. It also restores the fear learning and improves the fear memory in the fear conditioning test here. And it improves the unconditioned fear response. So these are important findings suggesting that this deficit may be reversible in DMD patients. And this paper was the proof of concept that we can compensate neurobehavioral impairments. But how can this therapy be improved? And why was the behavioral rescue only partial following DP427 restoration? So it could be because the dystrophin level were too low, and or because the adult treatment is too late. So we thought that we could intervene earlier at neonatal stages to see if we can rescue more phenotypes. So we decided to compare 2 delivery strategies: systemic injections to take advantage of a more permeable blood-brain barrier, and direct intracerebroventricular injection. For each strategy, we compare 2 therapeutic approaches, ASO exon 51 and also AAV-U7 exon 51, which is a vectorized sequences in the AAV-U7 system. But what is the AAV-U7 vector system? So a brief explanation. It's a technology mastered in our lab and published in 2009 by Aurélie, showing that modified U7 small nuclear RNA delivers antisense sequences to target pre-mRNA of the DMD gene. In this system, the antisense sequence is protected in small nuclear ribonucleoprotein particle complexes. Okay. And it localizes in the nucleus. This engineered U7 small nuclear RNA also modulates splicing to induce exon skipping and protein restoration very efficiently. And the AAV delivery enables a long-term expression, avoiding repeated injections. So first, we tried the systemic injection into the temporal vein of ASO exon 51. So we inject at postnatal day 2 into the temporal vein, and we follow the rescue at week 4, week 7, and week 14. And we obtain around 10% of exon 51 skipping at week 4, but this level was not maintained at week 7 and week 14. We also check the periphery, and we also detect some exon 51 skipping in skeletal muscle and in the heart. At the protein level, we could restore between 2 to 10% of exon skipping at week 4, but it was decreasing over time. So we obtain a transient effect, which is not sustained. And to maintain the rescue, we will need to inject again at week 7, but this will be concomitantly with the start of the behavioral study, which is not compatible with our therapeutic window. And moreover, this strategy seems not to be more efficient than ASO delivered in adults. So we switched to systemic injection of AAV-U7 exon 51, hoping that we can have a long-lasting effect. And as you can see, we obtain on only low level of exon 51 skipping in the central nervous system, around 5%, and a very high exon skipping in the heart. We checked also the protein level, and we couldn't detect any DP427 protein restoration, only in the periphery. So it seems that the systemic injection of AAV9 doesn't target efficiently the central nervous system. Okay, so we tried the bilateral ICV injection, which is a more direct injection into the central nervous system. We begin with ASO exon 51. And we were very happy to see that we obtain up to 60% of exon 51 skipping in the hippocampus, 70% in the cortex, and 30% in the cerebellum at week 7, but it falls at week 14. We also checked the periphery, and we were able to detect very low exon 51 exon skipping. And at the protein level, we can reach up to 21% of DP427 restoration in the hippocampus, 20% in the cortex, and 9% in the cerebellum, which is quite good, but it declines sharply by week 14. So a single injection of ASO is insufficient, and levels drop over time. So again, we will need to inject again at week 7, concomitantly with the start of the behavioral study, which is not compatible with our therapeutic window. So finally, we tried the bilateral ICV of AAV-U7 exon 51, hoping that we could have a long-lasting effect and a strong effect. And we were very happy to see that, yes, we can have a robust level of exon 51 skipping, increasing up to week 14. And we could also detect it in the heart, which can be interesting therapeutically because there is also a deficit of dystrophin in the heart. And at the protein level, we detect a significant level in all brain region, around 50% in hippocampus, 40% in the cortex, 20% in the cerebellum at week 14. And this was very promising for our future neurobehavioral studies in large cohorts of MDX52 mice. So what is the take-home message of this study? So first, the direct injection by ICV is more efficient than systemic injection for ASO or AV9. The neonatal ICV of ASO exon 51 leads to robust DP427 restoration in the brain, but restoration levels decline by week 14. So we have a limiting long-term effect. And finally, the neonatal ICV delivery of AAV-U7 exon 51 achieves high and sustained DP427 expression in the brain, which is a very promising for our ongoing studies on emotional and also new cognitive outcomes that we are developing, such as social behavior and cognitive flexibility. And more generally, we can say that AAV-U7 small nuclear RNA is a powerful strategy to achieve robust and long-lasting exon skipping in the neonatal central nervous system. So I would like to thank my unit director, Ophélie Goyenval, all my colleagues, Mathilde, Amel, Miki, and Olivier, who had worked on this project, and also our collaborator from Neuropsy, Cyril Vaillant. And I will be happy to answer to your question.
Thanks, Ophélie. Great results. So I come actually from the brain field, and then usually we have a similar question, but then the other way around. So of course, in brain disorders, the brain is affected, and we can treat the brain, and we can very efficiently with direct injections we can treat the brain, but very often there are also some other peripheral problems. So then the question is how to treat these kind of things, and I think you have the exact opposite. So and then always the question is, so what about combination therapy? So would it be feasible to do both systemic injections and direct ICV injections? And do you think that would be a viable strategy? And did you test it in the lab?
Yes, it could be very interesting to do that, to do systemic concomitantly with the central nervous system injection. We didn't do that for the moment because we prefer to study both separately, and we have other projects to test new AAV serotypes that could be injected systematically and goes to the brain, such as AAV PHP-EB. So it's something that we are going to prepare next. But now, yes, I'm studying the rescue at the behavioral level of this project.
And another question that I always have is, you are treating these neonatal animals, so you see that when you treat them at a later age, and maybe the phenotype cannot be reversed. But do you have any information on these boys? So is it that they have like some emotional problems, some learning deficits
Yes.
from the beginning onwards, or is it that something that they develop during life?
Yes, it's something that developed during life, so it could be more severe at adult age. So yes, as we can treat now by some AV therapeutics or others, we can treat the muscles, and people could have a better life expectancy. So we are studying now if we can also improve the quality of life of these patients in the future. So that's why we are studying that.
Yeah, and any information on whether cells are just not functioning, or whether like you have like cells are degenerating? So are you aware of that maybe in patient brains? And did you also look at pathology in your animals? And can you see whether this pathology is reversed after the treatment?
So in the first study we made with Amel Saoudi, we looked at behavioral impairments, and it was reversible partially, but it's a very good point. The first proof of concept that we can reverse some brain comorbidities. And we hope that with these new techniques and new tools, we can restore more dystrophin and maybe restore more phenotypes. And in the human, we know that they have similar behavioral impairments, like autistic behavior. They have high anxiety, they have learning problems, etc. So
Yeah,
it could be, yes.
Did you look at sections, so the brain
At
sections
section?
of these animals? Yes, brain
Oh, yeah.
sections where you look at dystrophin expression, these kind of things, and is
Yes,
it
of
also
course.
reversed?
Yes, we can relocalize the dystrophin in the brain, in the neurons, and in the glial cells also, because you have the dystrophin in neurons and also in glial cells, and everywhere in the brain and in the cerebellum. So that's why we use for the moment AAV9 to target all the cells, and also ubiquitous promoters to have a very large re-expression. And yes, we have sections. We are studying that also. So I didn't show that for now, but yes, I would do next time.
Yeah,